All right, let me share my screen. Well, thanks for being here, all 4 of you. This is my talk on spatial simulation. And it builds I'm working on for a long time. It's really probably 15 years more of work on how to build simulators for spatial modeling. There it is. So traditional modeling is non-spatial and deterministic. So you have, for example, some reaction network we have here. This is a metabolic reaction network in E. coli. And you track the concentrations of each species as a function of time. So it's ODEs, equations, and then here's concentrations function of time. And that's really a great approach in lots of ways, like we know from Roadrunner and Tellurium sort of stuff. Advantages of it, you have analytical theory, you have flux modes, but about control analysis, you have Jacobians, lots and lots and lots of mathematics really plays into this kind of modeling approach. It's really helpful and you get a lot from it. That's great. However, not all systems can be modeled that way. Some things are stochastic. So instead of having a single trajectory of your time course of a concentration over time, you have stochastics and molecules are discrete and they drop down one at a time. So also a trajectory is possible. So you can drop down this way or some other trajectory, and you have noise in the system due to stochastics. And the general rule of thumb is if you have n molecules, your variation is 1 over root n. So if you have 10,000 molecules, 1% variation doesn't really matter much. If you have 100 molecules, though, 10% variation is important. So for small systems like bacteria or for like within some organelle with low concentrations, it actually does matter a lot. So stochasticity can be important. Also, biology is spatial. And that's a good reason why a lot of biologists use microscopes, because it is fundamentally a well-organized system with spatial stuff happening. There's a variety of kind of fun pictures off the web and from colleagues, kind of shows lots of amazing structure in biology. So does space matter? There's only 2 reasons why space matters. One, if you are investigating spatial questions. This is E. coli and called the MinD protein. And it's back and forth over time. So it naturally oscillates back and forth. That's how the E. coli finds its cell center for cell division. Once it divides cell center, if it divides off center, then you don't get DNA in both halves of the cell and causes problems. So once it finds cell center, so here's a simulation of the same thing, starts out well mixed, then spontaneously organizes into a back and forth oscillation. The simulation obviously has to have a spatial component to it because it's a spatial problem. Lots of questions have similar spatial components: cell polarization, trafficking of proteins or other things inside a cell, crowding is an important aspect of cells, cluster formation of proteins, lots of things are fundamentally spatial. So need spatial modeling. Space also matters if you have if dynamics come from indigeneity. Here's a predator prey simulation. This is red as rabbits, green as foxes, or red as prey, green as predator. It turns out well mixed. So it's a well mixed system here, but it very rapidly self-organizes into inhomogeneity. So you get this kind of boom bust pattern, which really arises from the spatial dynamics. So what are simulators that are used for these spatial models? There's a wide range of simulators. There's kind of the main ones that are out there currently, ranging from atom sizes up to protein cells, usually mice cells, parts of cells, bacteria cells, eukaryotic cells. And here are the software used widely. I'll go through them one at a time. At the smallest size scales, 0.1 to 10 nanometers, is called molecular dynamics. Here we have READY by Frank Noe's group in Berlin, and he models parts of proteins. So not the whole protein, every molecule or every atom in the protein, but every kind of sub-portion of a protein. These little moieties of proteins. He leaves out solvent, but has sub-protein detail. Spring salad is a little bit more coarse. That has springs between protein chunks, so spheres. A larger size scale, we have Spatiocyte by Satya Arjunan. It models space as a very fine lattice, and every lattice site is either empty or has some protein or other molecule in that site. It's not like a lattice gas or some automaton, if you know those things. It's very, very fast for simulating, but challenging to do accurately because of lattice artifacts. But does a good job with it, and he's got to work out really well with good simulations, again, for small size scale, fairly small size, 100 nanometers. Next size up is a particle-based simulators, Smoldyn by my software, MCell, which is very similar, Pittsburgh, San Diego groups, and those two are both particle-based, so if each protein is one particle, a sphere or other particle. Also, there's Hetesim and Median, which are filament-based simulators. So both these do filaments, they don't do molecules, but they have actin, microtubules, that sort of thing. Next, I scale up 10 nanometers to 100 microns. There's MesoRD and URDME, both in Sweden. In this case, it's a coarse lattice with many molecules per lattice site. MesoRD is a cubical lattice, URD has an unstructured mesh, but similarly otherwise. And then also there's Virtual Cell out of the UConn Health Center. That was a PDE solver, so deterministic, but spatial with these kind of finite elements that get solved for. Virtual Cell is expanded now to have lots of solvers in there, but it was originally a PDE solver. There's also CompuCell 3D, which is a lattice-based multi-cell simulation. If you have any questions, by the way, jump in and ask questions. Okay, here's a summary of the smaller end of things. MCell, Smoldyn, EGFRD I not mentioned, Spring Salad Ready. EGFRD is not a software, but it's more a algorithm, which is basically exact within approximations. And a summary of everything that's here. This table, if you want, is at small.org. Simulators, HTML, also on Wikipedia at this point too. So kind of summarizes what capabilities are of simulators. So that's kind of the whole size scale range from microscopic to macroscopic. And I'm going to sort of either remove detail from microscopic ones or add detail to the microscopic ones and go to the middle size scale of Smoldyn, basically. So here's pictures of kind of Smoldyn size scale, particle-based or spatial Gillespie is called, which is the coarse lattice. So it doesn't matter, actually. That's an important question to know. If you have a ODE solver, deterministic simulations, here's the classic predator-prey result. You have nice oscillations. The prey is in blue. It precedes the predator. Predator booms when prey goes up. Afterwards, it causes prey to crash and so forth. This is a classic thing you see in ecology class. That's a non-spatial version. If you make it stochastic, you get kind of drift in these peak heights. And that's, again, stochastic here. So you get this kind of drift of the size oscillations. If you add in space, things change, though. You get deterministic and spatial, add stochastic, spatial, stochastic, spatial, and now most accurate of these is particle tracking and spatial, where you get a very distinct boom-bust pattern. But basically, the point is, yes, methods do matter and give different results for different methods. So it's worth kind of being thoughtful about what you want to use in this case. So my advice is the least accurate method possible. That's adequate to your problem. More accuracy adds more headaches, but does not add any quality because use the least accurate method you can use. Also, my pet peeves is a lot of people rewrite software that has already been written. And my advice is use other software, not have finished programs, don't write your own, and check the validation of existing software because often it's not as accurate as people say it is. So now on to my software, the Smoldyn program. Again, it's been around for a while, I guess almost 20 years now. My first paper on 2004, and published on it a lot since then. It's a particle-based simulator. We have particles that diffuse around a space, and they collide, they react. They can diffuse on surfaces. They can be reactive on surfaces. So here's a quick summary of what it does. Continuous space, fixed time steps. Molecules are spheres or points, either way, and membranes are thin surfaces. Molecules conceptually are very simpler. They can diffuse, they adsorb or desorb at surfaces, and they react upon collision. And then features, a lot of features in this program. It's accurate. I've done lots of validation. I'll show some later on. It's quite accurate, and it's quite fast. I'll compare it again to other software, and it's consistently much faster than other options. It has real-time graphics, lots of tools you can use for manipulating molecules and observing them. And then newest features are rule-based modeling, reaction networks, and it's free, open source, of course, and many more features too. So here again is that same min simulation I showed before of it starts out well mixed, then spontaneously self-organizes into this back and forth oscillation. That's a nice demonstration of the Smoldyn software. As min system generally has become it's an interesting biological problem, first of all, but it's also become a test system for different simulators. As one of its tests here, kind of showing that it actually works well. So what's Smoldyn used for? Again, it's been used probably, we're going on 20 years now, but about 3 or 4 or 5 papers per year get published using it. So it's not a huge number, but it's substantial enough that it makes a big difference in the community. It's used for 2 main classes of projects. For system modeling, that's what I first envisioned it as being used for. So things like dendritic spines and neural cells, E. coli chemotaxis. This is E. coli cell. Then it has receptors up here, and then this whole connection machinery over here, and a flagella, which is not shown here, but it'll spin. There's a motor here in green. It'll spin and drive the cell forwards. Min system, I just mentioned, it's the oscillations back and forth in the cell. Yeast cell signaling. There's a few projects on that by myself and Tim Elson's group, looking at cell polarization and information transfer and yeast cell signaling, apoptosis. So all these things are system models. Also been used, kind of hadn't anticipated at first, but because of its high validation level, a lot of biophysics projects. So crowding, the question is, how does crowding affect diffusion coefficients inside cells? How does it affect reaction rates inside cells? A bunch of work on that. Diffusion barriers. A colleague looked at how this little neck in the cell affects diffusion from here to here. Looking at diffusion in a, you can see a corral region. So have easy diffusion, I guess, starts here, easy diffusion in one region, and then it hops from region to region and has this interesting diffusion trajectory of sort of hopping from spot to spot. My colleague and I did DNA target finding. So how do transcription factors find their DNA sites? So that's a sampling of them, lots more publications as well on the website. Running it is pretty simple. It's you download it from smoldyn.org as precompiled binaries, so just plain use the file instructions and install. It's pretty straightforward, but it's not a GUI though. It's a text-based software. Documentation, there's lots of things. There's the manual, 200-page user's manual, has everything you need to know basically. There's also, doesn't say here, read the docs website. There's libsmoldyn manual, And if you really want to get into it, there's the code documentation. That's really just for me, though. And also, 3 papers I've written is one of the papers I've written that shows how to use Smoldyn. And then my main approach is looking at an examples directory. It's about 100 example files, example directory that shows pretty much how to do every single thing you can want to do. To run it, go to command prompt and type in Smoldyn and your file name.txt. I'll show you kind of a quick view of user's view of things. User writes a text input file. Here's the same predator prey simulation. So that's the whole file, pretty short, plain text file format. Goes into Smoldyn, so a black box, and out comes graphics and text output, which is saved to files on your disk, and then plot that into whatever you want, Excel, Mathematica, MATLAB, your choice for analysis. I'll do a quick survey of the input file. It's the same one again. You have comments. You mentioned that the graphics are, in this case, it's OpenGL graphics, and graphic iteration is 5 time steps. It's a 3D system. It can be 1D, 2D, or 3D in Smoldyn. Rabbit and foxes are my species in this case. Give the boundaries to the system. These are periodic boundaries in this case. Time start, time stop, time step, pretty self-explanatory. Display parameters, colors, and display sizes. Molecule lists make things a bit faster to kind of organize molecules into different lists. And then a very few lines give a kind of the essence of the simulation. Diffusion coefficients, in this case 100 is the 100 units, and then 3 reactions. Rabbit goes to rabbit plus rabbit, rabbit plus fox goes to fox plus fox, fox goes to nothing, and that's really it. Then you initialize with rabbits and foxes, 1,000 of each, uniformly in space, and then you output stuff to the disk files. So that's the whole file. I'll show simulations at this point. Any questions first? All right. I'll move this over here. And do you see my screen here? You see my?
Yes.
Great. So here, for example, is just plain diffusion. Super simple, 2 species, and they diffuse, spinning around. I see my molecules in space. I can go, stop, watch things diffuse. So that's how it is, that one. Surfaces be done different ways. An easy approach is plain to say, I want a sphere, or I want a hemisphere, or I want a disk. Also, you do triangle meshes, though. Here's a cell given as triangle meshes, and molecules diffusing inside this triangle mesh. So they diffuse inside this cell. And what's not obvious in Smoldyn is also pretty simple from the user point of view. There's a ton of work, and you make things debugged. Getting things not cross surfaces is actually a whole lot more work than it seems like it ought to be. But I'm proud to say molecules don't escape this cell. They stay inside as they should. So again, this shows that you can do complex surfaces. They also diffuse on a as a surface-bound molecule. So now it's a sphere. Again, it's easy to say one sphere. I don't want to give all the triangles for the sphere. It's to say one sphere at the origin, and now molecules on the surface of the sphere, and they diffuse on that surface. So you see it's all pretty fast, and things diffuse as desired on the surfaces. Also shows that the graphic levels. This graphics here is the high quality graphics with nice shiny stuff, and things look good. Molecules are spheres with shadows, that sort of thing. Often use similar graphics for faster operation, but you can do either one. Reactions. Oops. Where'd it go? There it is. This is the same predator prey simulation, but I added in grass in this one. I have grass in green, rabbits in red, foxes in blue. So this basic reactive system. So again, reactions occur when two reactions bind together. In this case, I have a Zerthor reaction. Nothing forms grass. It just grows all by itself. Fox plus rabbit forms two foxes. That's a biomolecular reaction. And then foxes die on themselves, so it's a human molecular reaction. But again, you get sort of fun patterns here of these from the reaction dynamics. The same reaction approach here is useful for doing crowding as well. So this simulation has red and green balls in 2D system. So it might be a cell membrane. And here there's no reactions happening of like A plus B goes to C, but here, when two red balls collide like I do here, they push each other apart. So the rule here is red plus red goes to red plus red, but moved apart some. And so with that, things will select crowding and non-overlapping things. That's sort of fun in simulation because these green ones tend to get stuck at the edges. Once I get there, they get stuck. In the corner, they get really stuck. But it's sort of fun to see what happens with crowding. Another sort of fun one is this clustering one. Again, this is a reaction algorithm. In this case, it says red plus green forms the green, but it sticks onto it at the edge. So you get this sort of dendritic pattern developing over time. All right. So a bit more of this. The way reactions work, again, A plus B go from C, some rate constant K, to reactions here. When they come within, we'll call it a binding radius, they react, they form product. And my challenge, again, there's lots of work behind the scenes, was how do you convert from the user's K value to an internal binding radius? There's a lot of work that's now solved, but that's part of it's behind the scenes. Later on, this product here might diffuse for a while, and then eventually it will dissociate into A plus B again. In this case, it separates them by a called an unbinding radius. Some fixed distance here. The reason for that is because they might diffuse apart and never reassociate, or they might come back together again and reassociate. And so by varying this unbinding radius, you can control the probability of rebinding versus diffusing apart. I've put a lot of work into accuracy for Smoldyn. So there's some validation graphs saying the solid line is theory, the dots are Smoldyn, and sure enough, it matches up with theory quite well. And then here's how much noise should it have. So actually the stochasticity also matches the theory correctly. It should be within these bounds here. Actually, not within, it should match the bounds there, and it does. Signal reactions, same thing. Here's theory, here's Smoldyn, same thing. And again, it matches the expected stochasticity. So that's accurate. Pseudo reactions. Here I have a hard sphere fluid. So these balls packed into a box. And I looked at the mean separation between neighboring particles. It's called a distribution function. And again, we have the black line of the exact theory. The red dots are Smoldyn and very, very close to agreement. So it shows that works well too. Example of one more algorithm on surface diffusion. So you have a bent surface here. How do you simulate that? Well, this particle here diffuses at some distance like that. Let's say, well, it can't go there. It's not on the surface. And so the algorithm is take this initial trajectory, move it to the surface by projection, and then fold it around at the edges and reflect off the edges as needed. And my validation again was run this in Smoldyn. I ran 300 tiny time steps in the gold dots, and said, yep, that works well. Now can I do just one giant time step? Notice the blue dots, and it works just as well too. So by being able to do long time steps, you can get high accuracy. You can get great efficiency and still maintain high accuracy. Another newish thing is protein complexes. Lots of proteins in cells are not just a single protein run by itself, but as a whole complex of proteins. So for example, in the yeast from our signaling system, ST5 binds to ST18 is ST4, and ST20 is ST11, ST7, FUS3. All these things have multiple phosphorylation sites. And so you have this huge list of distinct species if you list every possible combination and all phosphorylation sites and all the reactions. You get this enormous explosion of species and reactions. And so to deal with this, I added BioNetGen to Smoldyn, which is a rule-based modeling program that handles these things. Also added rule-based modeling with wildcards as well as my own invention. So for example, if you have a receptor can bind to a ligand, it can bind or unbind. Receptor can also bind to a messenger protein on or off, and can phosphorylate messenger protein. So you have fairly few reactions, fairly few species, but already just these few bindings give you this many possible species and reactions. How do you model them? Well, use BioNetGen. Here's BioNetGen rules, or use wildcard rules. So with wildcard, for example, L is ligand plus receptor forms the receptor. And the star means this wildcard, just like you have for your operating system, say whether bound or unbound. And star R plus M star forms RM star, RM star. So base head receptor plus messenger binds regardless of whether it's just bound to a ligand or not. Also regardless of phosphorylation state. And then L or M at the surface, or L or MP, so it could phosphorylate the messenger protein and then it'll dissociate on this solution. So basically, it's a very simple approach to give rule-based modeling and to give lots of reactions with fairly few statements. That's a fun project. It had lots of same examples of rule-based models, and all these reaction networks here I can model with fairly few statements with rules. Another somewhat recent thing is hybrid simulation. Particle-based cell stuff is good, fairly fast, accurate, but it's not as fast as lattice-based simulations. And so our new approach was, let's do adjacent space hybrid simulation of left side as particles, right side as lattice, and then molecules can smoothly transition between particle and lattice representations at this surface. Again, the validation, and it worked out well. Whoops. A few more things. There's a API for Smoldyn. If you want to connect to it through C or C++, that's now working well. Or if you want to connect through Python, also works well. There's now a Python simulation run in Smoldyn. So you import Smoldyn, import matplotlib, and you define simulation, add species, add solutions, so forth. So basically create a whole simulation in Python. I'll show up a few last things here. Let's see. Almost all the features came from users' requests. And so one of them was someone wanted to have moving surfaces. In this simulation, there's a cell, potentially, and some organelles maybe. And the organelles, or these, I don't know, my cells, or they are, can diffuse inside a cell. Also, going to stick to other ones. Oh, it's not going to stick very well. Like, anyway, it's called snare proteins. Snare proteins will bind between the two match ones. So if it goes over there, it might stick. Oh, almost there. No, didn't bother this time. So go ahead. Notice that on the surface, they have surface-bound molecules. They stay on the surface just fine. If one's inside, they stay inside just fine. the surface data is fine. I'm going to rerun this again and see if it binds this time. Yep, there it is. It's stuck. All right. That is diffusing surfaces. The request was for long-range forces. So I don't want just contact forces, but I actually want to have molecules that can attract each other across the whole simulation. And so here, the green ones attract. Oh, that's wrong one, actually. Sorry. I wanted long-range force 2. So green and red attract each other, blue repel each other. So there you go. Green and red have stuck together, blue have repelled, and are now as far apart as they can get from each other. And then another one I did a few months ago was a flow. Someone wanted to have a Poisson flow, and so I added in a general flow instructions. So now we can have things spin around and then bounce off the surface and kind of slide past it. So just kind of a bunch of fun stuff you can do with this part of the base simulation. And if you use Smoldyn, just a few last words of advice. There's a lot of parameter output. And at first, ignore it, doesn't matter much. But if you use it very much, you should read it and read the warnings and understand it, because there's a lot of stuff here. And it's a research tool, it's not just a user tool. So it's worth learning how it works. And last slide, more information. Here's a website for it, GitHub site, read the doc site, face papers, and ask questions to me. Lots of people put knowledge for financial support and moral support and help here and there. Particularly, I want to mention Dilawar Singh has done a Python interface and been very helpful lately. So that's all I have, and do you have any questions?
What was, when developing Smoldyn, what was your favorite part to design, or the most intellectually challenging, I guess?
I think the reactions, that's sort of a very early step. And I don't know what I was doing at all. I didn't know if it was this or not. It was just sort of a way out on my own, developing this whole new concept. But it was kind of fun, though. Also, I came to it, I was supposed to use MCell for my postdoc. I was assigned that task of using MCell to research. And I tried it at first, and I couldn't get it to work. Also, I knew it was wrong. I knew it was just plain full of just plain conceptual errors. I thought I'll try it myself as my own version, but done correctly. I didn't intend it to be a career, but there it was.
So was your postdoc very physics focused, or was it more in the realm of like systems biology, software?
It was supposed to be system biology. That was what I was hired to do. I actually did physics, though. I did coding and physics, but it's supposed to be more E. coli modeling.
I see.
Yeah.
I have a question.
Mhm.
So maybe on your page 8, you mentioned for different scales, there are different tools correspondingly. You have mentioned a little bit in your slides, but I wonder if you could talk a little bit more about, so for different scales, what's the major difference among those tools or among those scales?
I mean, different things are approximations. So like Smoldyn, its low end cutoff is a bit over a nanometer because it's accurate down below that in some sense, but it doesn't match biology anymore. Biology is complex protein shapes, and to it, other things a sphere. And so it's just plain, yes, its algorithms are accurate within approximations, approximations are no longer valid to treat a protein as a sphere below maybe 2 nanometers. At high size scales, Smoldyn becomes just impossibly slow at more than like 10 micron size scale. That just sets the size scales here for Smoldyn. And similar things for other things. Like Ready, it's more accurate for the protein size scales, so you can down to individual atoms if you want to. This goes down to here. But then it gets impossibly slow above 10 nanometers. So there's all these things are defined by where their accuracy cuts off the low end and where they become impossibly slow at the high end. So does that answer your question? Or?
Yes. I also wonder if there's any mechanism difference for different scales when you do the simulations, or
Yes. So these things up here are like different algorithms produced. So like PDEs are a gridded system and much more efficient, but it's quite a different method though from particle-based. So here's particle-based, Smoldyn and M-cell, and different approach, different speed scalings, and this is more accurate than PDEs, but also it, I guess, too slow at larger size scales. So yes, approaches differ quite a lot in these different approaches.
thank you.
Mm-hmm. More questions? All right. Stop sharing.
Okay. So if you don't have additional questions, I will stop record.